Um, Good morning, students. Am I audible?
Good morning,
Good
ma'am.
morning, ma'am.
I don't still see all the students. And probably this was your first class, right?
No, ma'am, we had a class before this.
Okay, you had a which class?
Some of us had environmental biotechnology, whoever opted for it.
Okay, fine, fine. I thought this was the first class. Okay, anyways, so we can start with this class. So today is the practical class, and the first practical which is being listed in this course is basically the demonstration of Koch's postulates in fungal and bacterial plant pathogen, right? So to this, we first need to know exactly what are Koch's postulate. So if we talk about Robert Koch is was a very well-known bacteriologist, and he had done a lot of work in the area of bacteriology. He did some inventions in the microscopy also, and he has done some work in anthrax and tuberculosis, then cholera, right? Many other things. So beside that, he also demonstrated Koch's postulate. So basically, what our Koch's postulate is that how do we know that particular organism or particular plant is infected by the certain pathogens? They could be sometime two or three pathogen also, right? So we need to at the end come to some conclusion that this plant is infected by only this pathogen, or there are two or three pathogen, right? So in order to prove the pathogenicity of any pathogen, could be a bacteria, could be a fungus, or could be a viral pathogen, we need to follow Koch's postulate. We need to, there are four postulates, and we practically need to follow those postulates in order to prove the pathogenicity of a particular pathogen, right? So during the late 19th century, bacteriologist, which is known as Robert Koch, he laid down a set of rules for confirming that organism is the cause of the disease, right? So these are now known as Koch's postulate. And when some plant it is infected by any fungus or any other disease, right? So there are chances that due to there could be another pathogen or another organism could be in a, can cause an infection in that particular organism or in that particular plant. So how do pathologists know that pathogen or particular pathogen has caused a particular disease, right? So we need to make sure that this particular disease is being caused by one particular pathogen or two pathogens at one time, right? So for that, we need to apply Koch's postulates to the disease. So this is basically experiment. This is experiment which has four steps, and we need to conduct this experiment in order to prove that this particular pathogen or organism is causing this particular disease, right? So according to the Koch's postulate, the first postulate states that the organism or a pathogen must be consistently associated with the lesions of the disease. Whatever lesions or whatever symptoms are there in the plant, so that organism should be associated there with that symptoms or those lesions or whatever symptoms it is causing, right? Then second says that the organism must be, or a pathogen must be isolated from the lesions and grown in a pure culture. So what does this mean? That we should be able to isolate that pathogen from that infected plant, since here we are talking about plant, right? So we should be able to isolate the pathogen from that infected plant, and we should be able to grow in a pure culture. So pure culture is very important here, that we don't want any contamination in the culture. The single pathogen in that pure culture should be grown. Only one pathogen, only one bacteria, only one fungus should be present in the pure culture that we are growing, right? Then the third postulate states that the organism or pathogen from the pure culture that we have already grown, it should be re-inoculated into the healthy plant or a healthy host, and then it should cause the same kind of a disease or the same kind of a symptom which was the originally observed first वाले postulate में जब हमने observe किया था, तो it should be the similar, it should produce the similar kind of a symptom on the plant, the similar kind of a disease should be caused. Then The fourth one is the organism must be, again, it should be re-isolated from the plant or from the culture जहाँ से हमने, say for example, we are working on the plant, so I have grown that inoculated that plant with the grown culture, and the similar symptoms are being shown. I need to re-isolate the pathogen again from that plant, and I need to grow into the culture, and it should be identical to the organism which is originally isolated, right? So ये four steps है this we need to follow. And how do we know they are similar? Because first step में when we are going to see whether it is a present, in the second step we are going to isolate and we are growing the pure culture. So if we are talking about the fungal culture, once we get this pure culture, we should see under the microscope what kind of a spores we can see under the microscope, right? Whether they are the similar as we have seen in the first case. Like what happened when we go to the field and we get the sample? The first thing in case of a fungal pathogen that we do is we cut the section of those diseased and the healthy part. We see it under the microscope, because if it is infected by a fungus, definitely some kind of a fruiting body, maybe some kind of a spores we are going to observe under the microscope, right? So that's how it will give confirmation whether it is some fungus that is infecting. That after the stage in the stage 2 also, like in the second postulate also, when we are growing in a pure culture, we should be able to see the similar kind of a spores which we observed earlier when we got the sample from the field, right? So under the microscope, similar spores, similar fruiting bodies can be seen. So similar structure should be available, right? Then D is the organism must be re-isolated from. Then again we need to isolate into a culture, and it's shown to be identical to the organism which is originally isolated. So in case of a fungus, we grow the culture in a potato dextrose agar plates, basically media, and then under the microscope we see, right? And then after that there are certain biochemical testing which is being done to know whether a particular fungus is present. In case of a viruses, this becomes little more easy, that we inoculate the plant, mechanically inoculate the plant, then we isolate the DNA or RNA depending upon the virus, and we can just directly go for the PCR and then the sequencing to finally confirm. So we follow the molecular steps. So in fungus also these days, we can easily go for the PCR or a molecular method detection, but these are the classical approaches that we used to follow when molecular methods were very less known. So to prove the pathogenicity of a particular pathogen, we need to follow these four postulate, and we need to prove them, and it take a lot of time. These seems to be four, but it like inoculating the plant, then isolating from a plant, and again re-inoculating and growing in a 25 degrees Celsius in a plate. So it definitely takes around 1 month to 2 months to prove these postulates at the end, right? So your today's experiment is basically to demonstrate Coates postulate using apples which are infected with brown rot fungus, which is Monilinia frutigina. It is a fungus which causes a brown rot disease in the apple. It is definitely a fungi. It belongs to Escomycota, and species, genus is Monilinia and species is Fruticola. So this one is Frutigena, right? So in this practical, again to determine which microbe caused that fuzzy stuff on your apple, we must be able to first describe the symptoms that we see on the infected apple and isolate the suspected fungus pathogen which is responsible for that disease. So we'll isolate the fungus in a pure culture. That means we'll grow the fungus on its own away from the host in an artificial media, right? So we are usually for the fungus, potato dextrose agar, PDA is the media which is being used for growing it artificially, right? Then use the fungus that we have isolated in pure culture again to inoculate the healthy apple. So whatever fungus that we'll isolate from the apple in the potato dextrose agar plate, we are going to inoculate that to the healthy apple. Right, then we'll record the symptoms that will develop on the healthy apple following infection with the cultured fungus. Also, your observations the same as recorded previously. So we need to see the similar kind of the symptoms or similar kind of the colony or a fungal growth on the plate is being observed that was observed at the first time. So this is how we are going to this, right? So for this, the material which will be required will be the apples, for example, in the picture second, and the third is showing the apples which is infected by this particular fungus. It is showing this kind of a symptom. This one is the healthy apple, right? So we'll need the apples which are infected, then cover slave slides and cotton blue stain to observe under the microscope, and immersion oils to make a pump. Sometimes when we view the things under the 40x, so we need immersion oils also, and then potato dextrose other media plus antibiotics. So these antibiotics are being used to avoid any kind of a contamination further, and then the fresh apples, right? So first practical, this particular practical is divided into 4 parts: session 1, session 2, session 3, and session 4, according to the course postulates, which are again 4, right? So first session will be provided with the infected apple with the fungus, and the 3 pairs of potato dextrose agar, which are supplemented with antibiotic to control the bacterial growth here. Then microscopic slides, cover slips, and a cotton blue stain. So first, what we'll do, we'll record the symptom of the disease. We'll examine the fruit very carefully, and whatever symptoms that we see, we are going to note it down, right? Then we'll cut through the apple and we'll see is there any color and texture changes in the infected fruit. For example, here we can see that infected fruit is a brown in color as compared to the healthy fruit. So we'll observe the symptoms using a microscope and compare them with the healthy apple. So we are going to compare, we'll observe the healthy apple also and the infected also, and the comparison will be made, right? Then using a sterile blade, we'll remove the pustule, which pustules are found on the apple. So these pustules, most probably they contain the reproductive structure of the fungus. That means it's different kind of a spores, right? So we are going to take that carefully and we'll place it on the microscopic slide. So that we can see it, whether there are some spores which are seen under the microscope or not, because with naked eyes we cannot see anything, right? Then yeah, okay, then we are going to observe what we see under the microscope. Then using a sterile blade, we'll isolate small piece of tissue from the infected apple and place one tissue at the center of the plate of the video. Similarly, this is for bacterial isolation. What we do, we usually streak, right? We make a solution of the infected sample, and then we streak or we pour. So in case of when we are isolating fungal pathogens from the live tissues, so we need to cut that tissue into smaller pieces, and that tissue should contain both healthy and the infected part. So I'll tell you later why healthy part also should be there. And then we should put it in the center of the potato dextrose agar plate, and the fungus usually we incubate at 25 degrees Celsius, and we'll just put it in an incubator for certain time, and after that we'll observe the symptom, right? So this is basically under the microscope the conidia of this particular fungus look like. So when we see under the microscopes, this kind of a picture will be obtained. And this one is the plate after we inoculate, and after maybe 4 to 5 days, we'll see this kind of a fungal growth on the potato dextrose agar plate, which again then we'll take out some part from this fungal portion, and we'll see it under the microscope what kind of a things we can see, right? Then only it will be confirmed whether it's the pathogen, same pathogen or not, right? Then in 2 session again, we'll have the isolation plates. From the session 1, we'll have healthy apple and all those things which we were having in the earlier things. So here we'll examine the plate cultures of the microscope of the microorganisms which are isolated from the apple, and we'll inoculate apple here. We'll inoculate fresh apple with the microorganism that we have isolated. So here we are going to use the pure culture, and we'll use that culture to inoculate the fresh apple. Then we'll sterilize the apple surface first. We'll clean it, wipe out with 70% ethanol, and then we can, we'll just take a piece of small fungal colony with a sterile scalpel and insert it into the healthy apple, right? So if time permits, so this not a time permit, this we should usually see that we should take a small amount of a fungal culture and we should check it under the microscope slide also. We should try. Now the apples will be incubated again 25 degrees Celsius, and then we'll follow the session. So this is how. we'll take healthy apple, not a cut one, and we'll puncture a little bit with the scalpel, and we'll put that culture in little bit inside the apple so that it get entry into the apple, and then we'll incubate the apple further, right? Then in the session C3, again, we'll have the apples which are inoculated in session 2 with the microorganisms which we have isolated from the PDA plate and other again the media and other things that are required, right? So here again we'll compare the symptoms of the disease in the inoculated apple with the recorded. So first session में whatever symptoms we observed on the apple, that those we got from the field, we need to compare those symptoms with this inoculated apple, which we have done artificially in the lab, right? So we'll see whether they are identical or there are any differences. And after that, we must re-isolate the microorganism from the apple that we have inoculated, right? So we need to prove basically that single pathogen is being isolated from the inoculated apple that we got the earlier, right? Now we'll move to session 4. Now, session 4 will be again provided with the re-isolation plates inoculated in the session 3. Again, we'll examine the culture, we'll observe, we'll take that culture, we'll see it under the microscope, we'll see whether same kind of a conidias or spores or fruiting bodies are observed or not. And after that only, if everything is fine, the same fruiting bodies are observed, same structure is conidias are observed, we can say that this particular disease is being caused by this particular pathogen on the apple. So here we are proving the pathogenicity of a particular pathogen, or we are proving that this particular pathogen is infecting this apple. Now we can say with 100% surety, we can say that this particular fungus is infecting or causing the brown rot of this particular apple or this particular plant. So this is how we prove the course postulates. It is a long experiment. It takes time. Sometimes we don't see symptoms. Sometimes symptoms are very less. Sometimes the fungal growth that we see under the microscope, we might not see very clearly the conidias and other things. So we need to be, sometimes we might need to repeat some steps in the experiment. So it's not that in one go we are able to prove the pathogenicity, or will be able to do the course postulate. Sometimes we might need to repeat the things. So this is how course postulates are proved, and they are very important whenever we are isolating any new fungus or any new pathogen from the plant. Since we are working on the plant, so we are talking about the plant here, right? So you all are going to write this practical in your practical file. We will mention, first you will mention about the course postulates, you write those course postulates, and after that you write the experiment the way I have written in the slides, right? Step-by-step procedure, right? And pictures, since you are not having pictures, so theoretical part you can do it. And if we come to the lab and we'll do practical, so we'll try to do this practical also. It's an easy one, though we'll need a pure fungal culture or fruit or some plant which is infected by the fungal pathogen, or we can go for a bacterial one also, right? So any questions for this practicals? So you can ask your questions. I have also uploaded a PDF form of this course postulate, so just go through that, and from that only I've made this today's lecture. It explains all the steps of a course postulate and how we follow the experiment, right? So you're going to write this experiment, and in next practical class, your experiment should be properly done in a file, right? Do it in a file in a proper way, because last time also there was a lot of confusion, and at the end when you were submitting the practical, few students, they were having some pages only. So either take isolated sheets only, separate sheets, write down your practical on those sheets, and then later you can put it in a file and you can get it submitted, right? Because still we don't know whether the classes are offline or online or in a hybrid mode. If situation is better, so definitely we're going to meet. But if situation is not so good, so I believe that we are going to have online classes for at least for a month or so, right? Because so let's see, by the end of this week, you'll get information from the university if they're continuing the classes offline or they're going for an online mode, right? So you'll get to know that, right? ओके, चलो फिर क्वेश्चन किसी को नहीं पूछना है तो फिर मैं पूछना स्टार्ट करती हूँ. तो व्हाट डू यू, आप लोगों ने क्या, व्हाट डू अंडरस्टैंड by this course postulates, and why do you think the course postulates are important and they are playing an important role in proving the pathogenicity? Huh? Yes, Devanshi.
Yes, ma'am.
अगर मेरा लेक्चर सुना है तो आपको पता होना चाहिए. पहले कभी सुना है आपने कोर्स postulates के बारे में बिफोर दिस लेक्चर?
Yes, ma'am. I had a PPT in immunology where I studied the coach postulates. Actually, I have a doubt related to these postulates because
चलो.
when he was discovering cholera, but cholera shows asymptomatic symptoms. So, how is it possible, according to its first postulate, that it should be isolated from infected person, not from healthy person? If we talk
सी
about cholera.
सी सी अब जब हम कोच postulates की बात करते हैं ना, तो दे आर ऑलवेज सर्टन एक्सेप्शंस जहाँ पे हम ये postulates 100% फॉलो नहीं कर सकते हैं, राइट? So cholera, cholera was, I guess, in 1883 पे there was some cholera problem in that area, and he found that the most of the people who died, they had certain bacterial infection, but they were not able to confirm at that time also that bacteria are the causative pathogens, right? So definitely in some ways, right? So course postulates are not 100% being followed in all kind of a pathogen. In viruses also, they are not 100% being followed, or in certain other kind of a bacterial pathogen also. So we are not saying yes, we need to follow the course postulate to prove the pathogenicity or to prove a particular pathogen is causal organism. But then there are certain exceptions always that they are not being followed 100% in all the pathogens, right? तो जरूरी नहीं है कि वो हो सकता है कॉलरा के केस में वो नहीं हुआ हो. सो इट इज नॉट इम्पोर्टेंट कि वो 100% फॉलो होगा इन ऑल द पैथोजेंस. वी कैन नॉट डू दैट, राइट? उसके भी ड्रॉबैक्स है. जो कोर्स postulates के जब हम ड्रॉबैक्स स्टडी करते हैं, सो दिस वाज द first point which was being raised, that how we can follow the course postulates in viruses, or how we can follow post postulates in those cases or those diseases jahan pe we are not sure that single pathogen is the causal organism. They could be the some other factors also which are leading to a particular disease. So definitely in those cases these are not being 100% followed.
Okay, ma'am. Thank you.
Okay. Okay, and then तो रॉबर्ट कोच का और क्या क्या रोल था? आई मीन कोच postulates तो उसने सिर्फ एक ही लेटर ही स्टार्टेड दिस, बट ही इज वेरी वेल नोन फॉर अदर थिंग्स आल्सो.
Ma'am, he discovered the anthrax disease cycle
या
and bacteria responsible for tuberculosis and cholera.
Ma'am,
और
attenuated vaccine.
यस, ही डिड दैट आल्सो. और माइक्रोस्कोपी में भी कुछ उसका रोल है. उसने कुछ इनोवेटिव टेक्निक्स यूज की है जो माइक्रोस्कोपी में, सच एज कंडेंसर्स का यूज कैसे करना है, या इमर्शन लेंसेज कैसे यूज करने हैं इन द माइक्रोस्कोपिक. सो सर्टेन मॉडिफिकेशंस इन द माइक्रोस्कोप एट दैट टाइम. He introduced those modifications for the better visualization of the pathogens under the microscope. So he did that also, right? Okay. So see, just go through these postulates one by one. And okay, so tomorrow we have a class, right? So let's do one discussion session tomorrow, that are Robert Koch postulates, I mean, can be totally proved, or what are the exceptions where these postulates are cannot be followed, or why those exceptions are there. So we can have one discussion lecture tomorrow, since tomorrow is our third lecture also. So we can have little discussion like last year we used to do in virology. So I'll just publish one assignment in one assignment section the topic for tomorrow's discussions, or one by one we'll have discussion on this topic also, that what are the postulates, what are the exceptions of the postulates, and in those exceptions then how do we, how do we prove the pathogenicity of a particular pathogen or a microorganism. So that discussion we can have tomorrow, right? So I'll just paste this in the assignment section, and please do your this practical files also, just submit on the time. And from me, that's all for today's session. If there's anything else we can discuss, please go for it. Okay, seems like you all understood this, right? So we can have the attendance, and then you're free for today's class, from today's class, right? Adhish Mishra,
Present, ma'am.
Ayushi Jain,
Present, ma'am.
Sam,
Present, ma'am.
Devanshi, Ishita,
Present, ma'am.
Janvi,
Present, ma'am.
Janvi Kshitij, Manaswani Namrata,
Present, ma'am.
Nandini Nehal,
Present, ma'am.
Rupal Sahiba Sanjana,
Present, ma'am.
Shakti Shivani Tanvi,
Present, ma'am.
Present, ma'am.
Tridiv Vivek,
Present, ma'am.
Gupta Yuvraj.
Present, ma'am.
Okay, so please make your practical files ready, and tomorrow we'll have one discussion session, right?
Present, ma'am.
Thank you. And
Okay,
yeah, I
ma'am.
heard that. Vivek.
Okay.
Okay, thanks.
Present, ma'am.
Thank you, ma'am.
Thank you, ma'am. Thank you, ma'am.